Hey Team Bio! Okay, so today we're going to be talking about membrane transport, moving molecules across a membrane. And this is important because if you're a cell, imagine, well, you are composed of cells, but it's important to move things into and outside of cells. So imagine we have positive things outside the cell. I don't necessarily mean positively charged things, but like food, for example. We need to get food inside of cells. How do we do that? Membrane transport. Additionally, there are going to be bad things inside the cell, like cellular waste, that need to come out of the cell. And there are also good things inside the cell that need to come out, like, for example, salivary amylase or catalase need to be secreted out of the cell. Additionally, there are going to be bad things outside the cell, like toxins, that we want to prevent from getting inside the cell. So membrane transport is all about getting the good things out, or bringing the good things in, keeping the bad things out, or getting the bad things out. How do we do that? That is the question we're going to be answering in today's screencast. Okay, so membrane transport. There are two major types of membrane transport. The first type is called passive transport, and the second type is called active transport. And the major differences between these two types of transport is that passive transport does not require energy, and active transport requires energy. And so, passive transport. Why is this so small? Okay, so let's start with passive transport. There are two main flavors of passive transport, and the first flavor is called simple diffusion. Okay, and to talk about simple diffusion, we really need to go back and look at our cellular membrane. So if you recall, we've got our phospholipid bilayer, and it's composed of phospholipid head and then our fatty acid tail. One of them is kinked. And I'm going to pause this here and create a membrane. Okay, so here's our membrane in place now, and as you recall, the kinked nature of this molecule gave the membrane a fluidity, so that things are always moving and diffusing around in the membrane. And this, if we look at this phospholipid, it might be over here, and then a couple minutes later, it's over here. So the membrane is fluid, constantly moving around, constantly changing, and as such, there are some times when there is a small micropore that opens up in the membrane because of this fluidity. And, no, I don't want to do that. So this tiny little micropore opens up, and teeny molecules, for example, this is an example of oxygen, O2, might just sneak right through this small micropore that opens up. And again, a small micropore might open up over here. Again, these pores are very, very, very small. And another molecule, this is CO2 here, might just sneak right out of the membrane. And so this is an example of simple diffusion. It's small micropores that open up in the membrane, allow for small and uncharged. Careful, the molecules here have to be small as well as uncharged, can move through. And the important thing to note about passive transport is that it's always moving from an area of high concentration to low concentration. So, in our cells, oxygen concentration is generally higher outside of the cell, and so it's constantly moving into the cell. And CO2 concentration, which is a waste product of cellular respiration, is high inside the cell, and so it's always moving out. So, simple passive transport is always moving from high concentration to low, and simple diffusion is just the movement of small, uncharged particles moving through the membrane down their concentration gradients through these little micropores that open up in the membrane. Okay, the second type of passive transport is called protein-mediated passive transport. So let's add another little section here. Okay, protein-mediated passive transport. And I'm going to add to this membrane. Okay, so I've added to our membrane, and you can see now here that there is a protein channel or a protein pore embedded within the membrane. And this protein pore is going to allow larger molecules or charged molecules to move through the membrane. So for example, let's say we have a molecule of sugar, which is much larger than O2. It can move through this protein channel or this protein pore in the membrane. I would just draw your attention again to the membrane itself. Remember that we have this hydrophobic interior. Hydrophobic interior, those long hydrocarbon chains are hydrophobic, and we also have our phosphate heads, which are very hydrophilic. And the dual nature of this membrane means that charged molecules do not want to pass through this hydrophobic section. Anything that's polar is not going to move through this very easily. So the important thing to know about protein-mediated transport is, again, it's always from an area of high concentration to low. So this sugar molecule, if the concentration is high outside the cell, then it's going to move into the cell. But if the concentration of the sugar is high inside the cell, it's actually going to move out. So again, passive transport is always from high to low, but protein-mediated passive transport is for large molecules or for charged molecules, because they can't move through the membrane easily. This is in contrast to simple diffusion, in which the particles are both small and uncharged. These can be either large or charged. Okay, so let's move over to active transport. I'm going to slide this over a little bit, give us a little more room. So the difference between passive transport, which doesn't require energy, and active transport is that active transport requires energy. Hey, how's it going? Okay, so a student just came in to talk with me, and I actually have no idea where I was, but I think I was talking about active transport and the fact that it requires energy. And the energy that the cell requires is ATP. This is the energy currency of the cell. Just like in the US we use dollars in order to perform transactions, the cell uses ATP. And so if we were to go from the United States to England, for example, we'd have to change our currency from dollars into pounds in order to make transactions over there. And the cell does the same thing. It receives energy in the form of sugars or fats and lipids, or sometimes even proteins, although proteins are kind of the energy of last resort for a cell, because they're the lowest in energy. The cell then has to convert those organic molecules into ATP, or transform the energy from those things to ATP. So ATP is powering active transport. And the first type of active transport that we're going to talk about is protein-mediated active transport. And protein-mediated active transport, much like protein-mediated passive transport, moves both large molecules as well as charged molecules. So large molecules, charged molecules, or small charged molecules. So protein, I actually don't like the way I set this up. I'm going to erase this. So the large molecules can be charged or uncharged. Or the molecules can be small and charged. And the difference between protein-mediated active transport and protein-mediated passive transport is that passive transport is always from an area of high concentration to low concentration, so things are always diffusing down their concentration gradient. Protein-mediated active transport is always an area of low concentration to high concentration. So we're using energy to push things against their concentration gradient. Now, something important to note about both types of protein-mediated transport, both active and passive. The thing to note about these is that we can control what enters or exits a cell with both types of protein-mediated active and passive transport. And the reason for this is that the protein pores can be closed, or the energy required to pump these things can be cut off. This differs from simple diffusion. Simple diffusion cannot be controlled because these molecules are just diffusing through micropores that open up in the cell membrane. We can't control CO2 from leaving or O2 from entering a cell, or just basically moving from their area of high to low concentration. Whereas we can control sugar entering or exiting the cell just by either closing these protein pores or even taking them out of the membrane entirely. So protein-mediated, both active and passive transport, the cell can regulate that. But simple diffusion, nope. Okay, the last type of active transport is called bulk transport. And bulk transport comes in a couple different flavors. Exocytosis is the movement of stuff that's inside the cell through the cell to the cell membrane and outside. And you actually, in your screencasts on catalase and amylase, talked about exocytosis. So it's the fusion of the vesicle with the plasma membrane to dump whatever the contents of that vesicle are outside the cell. So there's exocytosis, and then basically the opposite or the reverse process from exocytosis is endocytosis. So this is a cell bringing something in, and there are two major types of endocytosis. There's pinocytosis, and this is cellular drinking. It's or cellular sipping, and it's very small vesicles being taken off or pinched in the membrane. And then there's phagocytosis, and this is much larger particles being taken in. But on whole, bulk transport requires the use of vesicles. And so vesicles require a lot of energy. So the ATP comes into play here. Vesicle formation requires energy, and thus active transport, or this bulk transport, requires energy. And in addition, ATP is required to move things against their concentration gradient. So these are the two ways that energy comes into play with active transport. Oh darn, we can't see it all on one screen. Well, that's it. I'm bummed that we can't zoom out. Wait, I have an idea. Ta-da! Okay, now it's all on one screen. Very fulfilling. Okay, just look at all the knowledge you now have. Okay, that is it. I will see you all in class tomorrow.